Our first speaker this morning is Amanda Rowe, coming to us from the Natural Resources Department of Canada. And if you're ready, Amanda, I'll hand it over.
Great, thank you. And I really appreciate the opportunity to present from very cold and snowy Canada. We still probably have 6 feet of snow on the ground. I'm just going to share my screen here, and we should be good to go. All right, so today I'm going to present work that I've been doing in collaboration with Anna Turpelin, who's my postdoctoral fellow, who just very recently had a baby, so I'm presenting on her behalf. There we go. So because I know a lot of people are going to be talking about different aspects of the spotted lanternfly, I'm only going to provide a very brief overview of its biology, and then spend more time talking about the aspects of our project on cold tolerance. So spotted lanternfly is an invasive plant hopper that is native to China and Southeast Asia, and it has a very wide host range that includes a number of tree species in Canada that are important in our forestry context, but its main host is the invasive tree of heaven. And one of the things that many have worked on that are here presenting is this idea of host range. The nymphs have a very broad host range that includes things beyond just those host trees, but and then narrows down for the adults to a very select number of tree species and other agricultural commodities. So one of the issues and one of the reasons we're here today is that there's an impact of spotted lanternfly on a number of important economic commodities. So the adults and nymphs are heavy feeders. They suck sap from the stems and branches of their host plants, and they exude a really large quantity of honeydew, which can grow sooty mold on the surrounding vegetation. And this really does threaten a number of agricultural commodities, in particular grapevines, which forms a really important component of both Canada and the US agricultural industries. Now, the interesting thing with spotted lanternfly is that although it's naturally a slow disperser, it's actually an excellent hitchhiker. So as an adult, and the nymphs themselves can move and hang on to moving vehicles, that the adults also lay their egg masses on man-made surfaces that can get moved around the landscape just with people moving commodities or just moving vehicles. And this human-assisted transportation really contributes to that long-distance spread. One of the important aspects within Canada is a phytosanitary concern, and that is because we actually import raw lumber into particular areas in Canada for processing in our mills, and there's concern that these eggs can move on that imported lumber. And now that ability to spread really has been reflected in the distribution of spotted lanternfly in the US. I'm sure you're going to see this slide or this map frequently today or the next couple of days. This was initially discovered in Pennsylvania in 2014 and has spread to at least nearly 20 states in the Northeastern United States and continues to spread outside of these quarantine zones. And it's really reflective of that ability to be moved through assisted transportation. And the predictions and modeling of that spread have been fairly consistent with what we're seeing on the landscape. And one of the biggest concerns is that this spread may reach California by 2030. And that includes, for us in Western Canada, major grape growing regions. Now, predicting where spotted lanternfly will establish is an important aspect of risk assessment and risk modeling. And for Canada, the northern margin of the distribution is of really important interest, as is areas in the US. And that niche modeling is based on biological data, but currently for the northern limits and the northern range margins, that biological data comes from South Korea, which has another invasive population. It has it's another invaded range for spotted lanternfly. So we really begs the question of whether the biological data collected in South Korea is reflective of the reality that we see in North America for our own invasive spotted lanternfly populations. And so are those thermal limits in those different areas comparable? So that brings us to our specific question, is looking at the lower lethal limits and the cold tolerance of spotted lanternfly populations in North America. And asking the specific question, do we think spotted lanternfly will survive winter in Canada? So as I said before, those egg masses are one of the really motile or stages that can get moved around a lot. The adults lay those eggs in late summer, early fall, and these are the life stage that overwinters. So they lay their eggs and then cover them with that waxy coating. You can see in this picture that waxy coating starts white, but then oxidizes and starts to turn into sort of a dull gray mass, making it very cryptic to see, particularly on any sort of metal or vegetation or logs. That waxy coating does provide some protection. The total, we don't fully know its role in protecting the eggs from external stresses, but it does seem to play a role. Now we know the spotted lanternfly has been predicted to survive in Canada and that the hatch rate of those eggs are sensitive to natural winter temperatures. However, we don't know where that northern boundary is, so we can't assess what particular areas are going to be most at risk for having established spotted lanternfly populations. So we had two main objectives for this work. First, we want to assess the cold tolerance strategy, which is meaning basically describes how cold and low temperatures will kill that overwintering life stage, and what happens if we manipulate eggs. So if we remove the wax or if we remove the eggs from the egg mass, does that actually have an impact on the survival of or the hatch rate of the nymphs in the spring? And the reason we needed to do that is we needed to have that information to effectively design the experiments to assess the lower temperature limits. And so we worked with John Roth down in Pennsylvania to source eggs because as of right now, we actually do not have established spotted lanternfly populations in Canada. And so we needed to have a source, a reliable source of eggs. And these eggs were collected in the very late fall, early winter, allowing them time to acclimatize to those local conditions, to those fall and early winter conditions before we brought them in. We imported them into our quarantine laboratory, which is a level 2 quarantine facility, so that we could do this work safely in Canada. So as I said before, the first year we were assessing the cold tolerance strategy and the impacts of manipulating the eggs on the egg mass. And when you assess the cold tolerance strategy, really what you're doing is figuring out when death occurs. And when eggs get cold, they could die at the point that the ice forms, because inside the egg is liquid until it freezes. If the death occurs at that point of freezing, it would be a freeze intolerant insect. But if the death occurs prior to that point of ice formation, injuries may be occurring that have nothing to do with the formation of ice within the egg, and those would be considered chill susceptible insects. And then we also just looked at what the impact was just separating the eggs from the mass to know whether we could use individual eggs for future temperature treatments. And so to measure that point of freezing, we measure what's called the supercooling point. So when ice forms, it actually is an exothermic reaction, meaning it releases a little burst of heat. We are able to actually measure that little burst of heat. That point, that temperature at which that occurs is called the supercooling point, the point that the liquid water within the egg rapidly changes to ice. That ranges from about -17 to -27 degrees Celsius with a mean of about -25. So it means that these eggs are able to stay in a liquid form very cold, under very cold temperatures. Now, we did also recognize that eggs that had cooled too close to that supercooling point, but hadn't experienced that release of heat, still failed to hatch. So that means that they are dying from injuries prior to that supercooling point. And so we were able to identify that spotted lanternfly are chill susceptible. For insects like this, it means that you need to do a much larger experiment and cannot entirely rely on the supercooling point to assess those lower thermal limits. The other thing to point out here is that the data points here, you can see the different colors. Those refer to individual egg masses. So we see pretty tight correlations among very, very similar supercooling points within individual egg masses, with a few outliers. We also found that removing eggs from the mass, so removing the wax and pulling individual eggs off of the egg mass, did negatively impact hatch rate, which means to do any further work with lower lethal limits, we actually had to work with the whole egg mass. It means we need a lot of egg masses to be able to do this with a reasonable replication. And so again, this is where having John provide us eggs from Pennsylvania was critical to this work. So as in year one, for year two, where we looked at the full full lower lethal limit experiment, we allowed the eggs to acclimatize in the field. Then we brought them into our quarantine facility and exposed them to different time and temperature exposures. So we took several different temperature exposures, ones that were short that might reflect temperatures experienced over a nighttime, so for 1 hour for 12 hours. Then we also did long-term temperature exposures, 10 or 15 days, which might be more reflective of a long cold spell, like a polar vortex event. And so this gives us sort of two different perspectives on the cold. Based on our previous work in year one, and then in the work published out of South Korea by Park in 2015, we were predicting that we would still see hatch for those short periods of time up to about -20. But then anything colder than that is where we would see, we would have no hatch, so that there would be significant or complete mortality at those lower temperature limits. For the longer times, again, we would still see hatch at the warmer temperatures, but then start to see impact at those colder temperatures. And so now what did we see when we exposed these egg masses? So we actually saw surprising survival at very low temperatures. So for the first one, we found that we had egg masses hatching at -25 after 1 hour exposure. So at the 1 hour, you can see we have egg masses hatching here. We even had hatching for egg masses exposed to -20 for over 10 days straight. So this is 10 days of constant -20. We still had eggs hatching after that exposure. And what was really interesting is that even in that very cold -25 exposure, we had an egg mass that had nearly complete hatch. So it wasn't just one or two individuals that were capable of hatching. We actually had a 100% hatch rate, fully successful hatch of this egg mass. And so this means that if that egg mass had been moved into a really cold climate, even if it experienced a -25-degree cold snap, you'd still get a lot of individuals coming out because these egg masses have quite a large number of individuals per egg mass. And if you see here in red, these are areas where we had predicted to have minimal to no survival for these cold exposures, based on what we had seen out of the South Korean data. So it suggests that the North American spotted lanternfly are in fact more cold tolerant than previously described from the South Korean invasion. And so this has important implications in assigning or assessing the risk of where spotted lanternfly will be able to survive and establish along that northern margin. And this is being used to help inform the risk assessment models used by our plant health agency, the CFIA, the Canadian Food Inspection Agency. And it also raises some interesting questions about why we see those differences between South Korea and here in North America. there is potentially we have regional adaptation, whether it's through selection, or we see plasticity in the response of the eggs to that acclimatization period in the fall. And it does suggest that this northern margin, may shift further north than what was predicted based on those initial niche models. And so the question about regional adaptation and plasticity is work that we are currently doing in collaboration with folks that are here on this call, as well as folks down in Lee Boardman down in Tennessee, trying to understand what might be influencing this cold tolerance. And so what does this mean for Canada? Well, spotted lanternfly is really sitting on our borders and is waiting to come in. We know that it moves. We know it moves really easily. And we know that we've got a lot to lose. We have a number of areas with large wine growing communities and a significant investment both from the commodity perspective, but also from a tourism perspective. And we can see repeated incursions every year, and these incursions seem to be increasing. In 2023 and 2024, we've had a number of incursions, both on the Peace Bridge, actually flying into someone's car, on plants that are bringing in commodities from the US. But more interestingly, we actually have these observations in the middle of the ocean or in Nova Scotia. These are actually associated with cruise ships. And so these bugs are actually coming in and hitchhiking on cruise ships, which is a really interesting, somewhat undocumented pathway. And so really the risk of incursion in Canada is high, and we are expecting to see more interceptions and are constantly on high alert for any potential establishment, and are doing a lot of outreach to be able to encourage reporting of sightings to the CFIA. And so with that, I'd just like to thank all of our collaborators that have helped make this work happen, and I'd be happy to take any questions.
Thank you, Amanda. There is one question in the chat, and we do have time for that. How well do eggs survive if frozen at a mild temperature, given that eggs are often laid on the sheltered underside of trees? I assume there will be more moisture near the eggs and a higher change of inoculating freezing. So I think the question is, is there an impact of moisture as well as temperature?
Yeah, and inoculative freezing is a fantastic question, and Lauren, we got to work on it. Let's just say it's on the to-do list. It's a we don't know at this point, but something worth exploring. And Gary, yes, thank you for mentioning it. I'm glad that we've been watching for them on cruise ships. I think it is certainly one of those unusual pathways, and one that certainly is important.
We have a question here from Wendy about whether the temperatures were consistent or average. So in the lab, I guess, are we looking in the lab here?
So Wendy, the work that we had done is constant, and that is completely artificial. It's elaborate. Like, we know that insects aren't typically exposed to constant temperatures, and the whole what would happen if we put these under a fluctuating temperature is a really interesting question. Again, that comes back at those ideas about plasticity. How are the insects able to respond, or those eggs able to respond to short-term temperature changes? And we don't know at this point. However, some of the work that we have ongoing in the field right now, actually we have working with Scott out of Chicago, we have egg masses that have been going through winter right now. We'll start to get at some of those questions about what real winter outdoor field winter temperatures actually does to that hatch rate in different places throughout the distribution.
All right. Well, thank you, Amanda. And if you like, you can add some, repeat that answer in the written form. But we will
I can,
move
yes.
on now to Melody Keena. Melody is a researcher for the U.S. Forest Service located in Connecticut, but this work is looking at tolerance of SLF in climatic conditions in California. Melody, are you there and ready?
I'm here. I'm going to share my screen.
Fantastic.
All right, and it popped over onto a different screen. All right. So this is some work that we've actually been doing for a few years. I first looked at how spotted lanternfly would do if we had them end up in Napa, California. And Napa is not very different from some places here in the East Coast where spotted lanternfly already is. So in conversations with people in California, we decided to do work with the Stockton temperatures, which are generally warmer and drier than most of the places here on the East Coast where spotted lanternfly has already been found. So major reason, of course, for this is that California's grape growing industry employs a lot of people and generates a huge income. So there's a lot of economic involvement in this industry, not only from the products, but from tourism. And so California, of course, is worried about what spotted lanternfly can do to grapevines since they like them and can damage the vines as well as damage the products. So some work that I published recently showed that although the thresholds that we developed for spotted lanternfly using constant temperatures would suggest that these temperatures in California wouldn't be good for them because we were seeing some deleterious effects even at 35 degrees C. We decided that based on this work that if you offer the temperature with a alternating regime as opposed to a constant regime, you can see that spotted lanternfly can survive and develop at temperatures past their upper threshold as long as it's in this alternating situation. So just for reference sake, the egg masses that we used for this study were sourced from across the range in the eastern populations from 3 different plant cold hardiness zones, and compared to the zone where we were mimicking, which is a 9B zone, these were sourced from 6B, 7A, and 7B. And to create this regime in the laboratory, I took the temperatures that were actually experienced in 2022 and took the maximum and minimum temperatures and then ramped the chamber between those temperatures on a daily basis. And you can sort of see the chart down in the corner. It makes it kind of look like a flower. We are also changing the humidity and the light cycle to match the climate as well. This was all done in a large chamber using larger cages for the larger instars. And we were using both Tree of Heaven and Concord grapevines, so they had a mixed diet. In the first generation, they developed quite well, faster than they would have under, say, a constant 25-degree temperature. Mortality was sort of to be expected. There's a little bit more mortality in the first instar and in the fourth instar, but still overall quite good survival. And this is the phenology that we saw. We started them in this regime in March, and we had first eggs in August, what would be equivalent to August 23rd. And then we had a little bit of precocious hatch in November. And the heat wave that we simulated is in this red box here at the bottom. The upper temperature for that week was 40 degrees C and the lower temperature was 20. And you can see from the upper graph that there was no apparent mass die-off of adults or stoppage of egg laying during that temperature. The adults lived a fairly good length of time in these temperature regime. The only case where we saw a little bit less survival was for 6B females. They didn't seem to survive quite as long in these warmer temperatures. but the longest surviving insects at this temperature were 101 and 102 days for both sexes. We also looked at egg laying under this regime. Of the 60 females that we were able to follow, because it's hard to have enough plants to keep them all, we had 20 of them that didn't lay any egg mass. We had the rest of them laying one or more egg masses. First egg masses were laid by 40 females, 2 egg masses by 19 of those, 8 females laid a third, and 3 laid a fourth egg mass. The first egg masses were laid about 50 days after the adults had closed, and then after that, it was about 1 to 2 weeks for the next egg mass to be laid. And those egg masses started hatching in March, which is what I had predicted to set up this study to start with. So the timing of hatch was following what I've seen across the range here in the east and been able to predict that way. And it was fairly synchronous, as you can see here too. Some other factors about the hatch: first laid egg masses tended to hatch before later laid egg masses. This has been something I've suspected, but because in the field there's microclimate differences between where egg masses are laid, this has been hard to get at. But using this regime, we were able to see that this is indeed the case. And we've seen indications that this might be the case, that microclimate is playing a major impact. We followed egg masses hatching at a site in Fairfield, Connecticut that were kept outside the whole time. And those that were laid on the undersurface of rocks hatched before those that were laid on tree bark. This was one of the only temperature effects that we saw on these populations, was that for that 1 week that the high temperature was 40 degrees C, any egg masses that were laid before that time, so they would have experienced that temperature, or were laid during that time, tended to have much lower hatch than those that were laid later, although there's variation in hatch across the time period. We also were able to look at first laid, second laid, third laid egg masses, and we didn't see any appreciable difference in the number of eggs that they were laying in each of those egg masses. We then decided to keep going with this experiment. So we took the hatch from that first generation and grew them up to adults. And this is the results. We started with 564 first instars. We had about 28% die, and we had to kill off about 14% because we didn't have capacity to hold all of them. We reared through 151 females and 180 males that we used for a mating study, and I'll give you a little bit of the results of that. Average weights at emergence were 0.18 and 0.13 grams, which is probably close to what they would be in the field, although nobody's out there actually measuring what they weigh when they first come out. We started our mating study well before we expected that there might be any mating occurring, at the point when most of the males and females had emerged. And we indeed did not see any male fluttering until quite a bit later. Male fluttering actually occurred when the light cycle, which you can see on the bottom, reached the point that we were just starting to go below 14 hours of light. This has been seen and published on in rearing research previously, but this is another case where that seems to be the switch point, and it looks like it may be the switch point for mating. And so then we followed the matings. We observed each mating, and you can see the matings, and then you can see the egg masses being laid over time. And here's another indication that that is the switch point. If you look at the age at which the individuals mated versus the Julian date that they became an adult, earlier adults waited until they were the age at which that 14 hours switch point occurred before they actually mated, while those that were later adults were able to start mating at an earlier age. And this was a very interesting finding that also supports this idea that mating is cued by the onset of lighting coming to and being slightly below this 14 hours of light. And then we also were looking at time from mating to oviposition. And we were able to hold the individuals in large cages. And some points in time, we had only one day of mating in that cage. And so we could get sort of the bounds for this. It was about 5 days to 22 to 25 days, somewhere in there, that the first egg mass was laid after the mating occurred. And then you can see this, as this graph shows, that there were later laid egg masses. Those would be the second or third laid egg masses by individuals. In the mating study, we also found that the average time to first mating in this regime with this lighting cycle was 51 days for females and 61 days for males. We observed that all males lost their claspers. This is a part of their body that helps them stay connected to the female to complete the mating during the first mating. And in fact, we saw males and females having extreme difficulty separating in some cases. This also played out that males that had lost their claspers were able to subsequently mate, but the matings lasted for a shorter period of time and were in some cases less successful. You can see that first matings lasted about 2 hours, whereas all subsequent matings were only about an hour long. And we observed males mating up to 4 individual females. This work is all, this mating study is all being done with Claire Rutledge at the Connecticut Agricultural Experiment Station. So, summary. That week-long heat wave of 40 degrees Celsius maximum temperature and 20 lower temperature didn't have a significant impact on spotted lanternfly survival. The only impact that we saw was a possible effect on egg hatch for eggs that experienced that week. Oviposition occurred during the same time of year as it does for lanternfly here in the East Coast, which has already been established. But we also were able to see that 14 hours of light seems to be a switch point for mating. We had some precocious hatch that occurred, but we didn't follow these first instars, so the fate of them in a natural setting remains uncertain. Adults lived a relatively long period of time. Females laid multiple egg masses, and males were able to mate multiple times. Hatch started in March for this regime and matched the predicted time for this study. Follows the same heat requirements as eggs here in the East Coast. And progeny were quite viable under these conditions and grew very well. In fact, not a bad regime to use if you want to rear this insect in the laboratory. Of course, there were a lot of people involved, both in collecting egg masses and following all of these insects, watching these matings, etc. And this work was funded by the Forest Service, although we've got some additional funds coming now from the Farm Bill to continue some of this mating study and other work. And with that, I will take questions.
That was
Right.
Thank you.
Yeah, I see the first question is there potential for 2 generations or more per year in the state of California? Probably not, because of this light cycle requirement for mating. And even if those precocious hatchers, which were few in number, were able to grow, they would grow at very slow rate because the temperatures would be pretty cold for them through the California winter. And so they might end up joining with the March hatch and being just slightly early from that. So I don't think so. Do you think this could be a potential problem for other states in the East? This also shows that southern eastern temperatures probably are not going to be a problem for this insect. And what does Julian date mean? Julian date is the number that corresponds to the day of the year. And then we just extend it when we wrap over to a new year.
Not seeing any other questions, so thank you so much, Melody. Our next speaker is Matt Helmus. Matt is with Temple University. He is one of our main modelers for this insect. And he will be giving us a framework for invasive species data science. Matt?
Great. You can hear me okay, right, Jacob?
We can, yes.
Great. Okay. Well, thank you, everyone. So today I'm going to talk to you about some work that we're doing with modeling and focusing on stakeholder apps and risk models for spotted lanternfly management. And so the work I'm going to be presenting is really the work of three main researchers, Jocelyn Bem and Benjamin Seibold. We all do different aspects of studying spotted lanternfly, but really a lot of the work that we have been doing is from our team of undergraduate students and research techs, graduate students, and postdoc researchers. And so whenever I talk about some of their research, I'll have a picture of them up in the corner. And so just quickly an outline of what I'm going to be talking about. I'm going to be talking about four main topics, and these are sort of the take-home messages I'm going to give you right now. The first is we've been studying spread, and we've been finding that diffusive spread for spotted lanternfly is slowing, but the long-distance jump dispersal spread is accelerating. We've been identifying specific property types where surveys are most likely to find a new population of spotted lanternfly. And then we've been looking at 4 areas where spotted lanternfly is already established, how climatic variability can increase the likelihood of spotted lanternfly outbreaks. And then we've been thinking about modeling vine thresholds where growers can then initiate control. And what we've been finding is that those particular vine thresholds are a bit variable, and they're affected by how spotted lanternfly is moving throughout the surrounding vegetation. But I'll go through each one in detail and hopefully you'll be able to have these take-home messages clear in your head by the end of the talk. So first, we've been asking this question of how fast is spotted lanternfly spreading? And we put together a big database of spotted lanternfly surveys from all of our state collaborators in the USDA. And this is an open access database. Anybody can download it. You can go to the GitHub website that's there and download these data and use them for yourself. And so first, what we try to do is we try to estimate diffusive spread. So diffusive spread is just sort of like that spread that you think of, like a drop of water or something sort of like spreading out, on a landscape. And we know that diffusive spread rate is expected to be faster whenever you have these surveys, which are just, GPS points, whenever you aggregate them at coarser resolutions. And that just sort of makes sense. If you're aggregating points at a very, very a small resolution, then the total area of the invaded range is going to be smaller than if you aggregate it at larger areas like, states or counties or something like that. And so do we see this with spotted lanternfly? And the answer is yes. So we're finding that spread rate is faster when you look at different resolutions of the data. And this is sort of a statistical artifact that you have to account for. So here, what I have is total area invaded on the y-axis, and I've got time or year on this x-axis here. I've got a bit of a typo there, so don't ignore that. Each one of these different panels here is the resolution that we've aggregated the data. And as you can see, as you increase in this coarseness of the resolution, our spread rate is increasing from about 10 kilometers per year, we're estimating across the full time series, all the way up to about 35 kilometers per year. We also know that for a lot of invasive species, diffusive spread rate is expected to vary over time. And so you're expected to see sort of a lag phase where it takes a little bit of, it's kind of slow spread. Then you get a really rapid spread, and then you get a saturation or a slowing of the spread, sort of this S-shaped curve. And so do our spread rate estimates, do they follow that S-shaped curve? And the answer is yes. And so this is at the 2 kilometer grid here. That red line is that S-shaped curve, and that's fitting the data better than this straight blue line. And so we are seeing diffusive spread rate slowing in the most recent years. And this is true across the different grid sizes. This is the 50K grid and the 2K grid. Both of these are showing the exact same pattern. Now, I don't have time to talk about it here, but we developed a way to optimize the right resolution that you should be looking at. So those of you who are working on spotted lanternfly data, say in the States or in Canada, et cetera, we have a method that's in our paper of how to optimize the right resolution to look at. And what we found is that using this optimized resolution, we found that the highest spread rate was estimated to be in 2017 at about 21K per year. But since that time, now in 2023, the last time that we looked at these data, then it's decreased by about 80% to about 4 km per year. And so we're seeing this decrease in this spread rate in these most recent years, which is really interesting. And so the question really is then, how much of this slowdown in the diffusive spread rate is due to management? Because there's been a lot of management that's gone on here. And so that's really an open question that we need to address. But we know that spread isn't just sort of that diffusive spread, but there's a lot of these long distance jump dispersal, accidental transport events that occur as well. This is, for example, is that transport event, from the main area down into Virginia. That was sort of the first long distance one that we saw in the United States, for example. So here, what I'm going to show you is just the time series of the spread when it was first found in Berks County, Pennsylvania in 2014. All the points here are locations where spotted lanternfly was found in that first year. And then I'm going to show you how we've identified then jump dispersal events. So this is 2014 here. This is 2015 here. So it's, when they actually did this survey in 2015, it had spread a lot farther than in 2014. In 2016, it had spread out across multiple counties here. And then in 2017, we started to identify significant jump dispersal events here, both clusters of points and then isolated survey points as well. We did this by developing a statistical framework using null models and a lot of randomizations to try and estimate and partition spread into the diffusive component of spread, and then this long distance jump dispersal component of spread. And this has been submitted for review. So in 2017, we saw sort of only about 2 jump dispersal events. 2018, we saw a lot more, including that Virginia population that popped up. 2019, we saw even more. Here in 2020, now we've zoomed out on the map. We're seeing more in 2021, 2022, et cetera. And what we're finding then is that we're seeing this slowing in diffusion or even a constant diffusive rate, but spread is really now being driven by these long distance jump dispersal events is what's happening. And so through time, just looking at the diffusive component, that's that dark gray box plot here. You can see that's, more or less, staying the same or constant based on this analysis, but that when you include jump dispersal, we're getting these massive increases in sort of that invaded area, in the invaded radius, that range expansion that's occurring. So here, what I've plotted is the number of jumps on the left here, panel A, and then on panel C here, the distance of each jump from the core. And so we're seeing both jump number and distance increasing. And so here we have, the number of jumps through time, and it's been increasing, for example. And the 2 different colors here has to do with sort of isolated points versus clusters of points. It's not that big of an issue. And then here, if we look at the distance of each jump from the core, these later years in 2021, 2022, those are the years that are way out here that are really, really far from the core, where you see some of those earlier years being much closer to the core. And so we're seeing that jump number and distance increasing through time. And so I've showed you now that we've had these jump dispersal events that we can identify. Are there then, particular property types associated with these jump dispersal events? And these property types can be termed invasive species transport hubs, where they're properties where the convergence of vehicles, goods, and people facilitates this jump dispersal that occurs. And so all these different points here, these jump dispersal points, can we identify the types of properties statistically that they're associated with? And then can we use that statistical association to then try and inform management on where they should go and search for new populations? And the answer is yes. So we're able to identify the types of properties. Using randomizations and null models, we found 8 different types of transport hubs that were significantly associated with these jump dispersal events. There was a handful of property locations that we found that weren't really statistically associated with these transport events. We used 2 different null models, one that was sort of just a full randomization of all the data, and then one that accounted for a proactive search that is typically done whenever surveyors from states or the USDA are going out and looking for new events. So we also developed another statistical model there. We found that shipping distribution centers are quite strongly associated. Truck rental companies, stadiums, flea markets, which is a little bit surprising. So these are areas where people congregate. Landscaping businesses, truck stops, amusement parks, and then railroad stations as well. And this railroad stations is kind of expected because there's been a lot of talk about how, railroads and hitchhiking on rail cars or on the goods that are shipped in rail is one way that they're spreading. So here, for example, this is a location in Altoona where one of the first jump dispersal events were found in this part of Pennsylvania. And so this is a huge number of rail lines that are right here. And so then we also asked the question of, are these jump dispersal events associated or close by these high density of rail lines or the high density of roads as well? And we find that if we look at the smaller secondary roads, the primary roads, there's not really any statistical relationship with these jump dispersal events. But with rail, those areas that have a high density of rail are strongly associated with these jump dispersal events. They're very high density. They're very close by to these jump dispersal events, as kind of what we expect to see based on a lot of field observations. And we put all this information together in apps, in mapping applications. And we've done this for a lot of US states and a lot of organizations as well, where you can zoom in and zoom out at different locations and try and identify particular transport hub locations that then you can go out and survey and try and find these jump dispersal events. And so we have about 42 of these apps across 24 states right now. And then we're developing more. And if your group or your state wants an app, please do reach out to us after this talk, and we'll develop one for you. So we've been talking about spread, but now we're going to talk a little bit about established areas. And we've been looking at how climatic variability can affect SLF outbreaks. We've done this with doing population dynamics models. And so we developed a system of partial differential equations that models the life cycle of spotted lanternfly based on a lot of the information that was presented in the previous two talks, for example. And then we developed a framework where we looked at population growth under a very constant environment versus one that adds stochasticity to it. And so this is what I mean by that. So here, for example, is Napa, this is Detroit, and this here is Atlanta. And if you think about sort of this season, you sort of have sort of, it's colder in the winter and then warmer during the summer, and then it gets colder again, sort of that curve, right? And it's different across these different climatic areas. But lanternfly doesn't really experience sort of a very smooth mean temperatures. They experience sort of day-to-day fluctuations or variability. And so we compared our population growth models both in the constant case versus the variable case. And we asked, does that affect growth rate? We did that across locations across the United States. And I'm just going to take two, for example, and go through these examples of Napa and Yosemite, which are very close together, that show then how small differences in these temperature regimes can have really large differences in whether or not you see outbreaks. So what I'm showing here then is a figure. On the y-axis, this is the growth rate of a population at either Napa or Yosemite. And then I'm going to increase temperature variability. I'm going to make the temperature more and more stochastic. This line here is the temperature, is the growth rate under mean seasonal temperature. And then this is the equilibrium growth rate. Underneath that value, populations will decline. And so then if you see a point that's up here, it means it has higher growth rate with variability. Points here has lower growth, and then points here is in decline. This is Yosemite. These are the variables. As you increase temperature variability, pretty soon populations start to crash. And so temperature variability pretty much causes, at least in Yosemite, these populations to crash. But this is Napa. And what's happening here in Napa is that Napa is in this really sweet spot of population growth for lanternfly, where in some years it's just really great, where lanternfly just hit all the right developmental markers and the temperature is just perfect, where you can have huge outbreaks in the population. And it takes a lot more variability, as you can see here, compared to Yosemite, for the population to crash. Now, if you, in the model, turn off diapause and you say, you cannot diapause, let's say that the population has evolved to not diapause, we see a huge crash in the population. And so what this means to me, or at least suggests, is that we likely won't be seeing multiple generations per year in California, to get at some of the questions that were asked in the previous talk. These now are all the counties in the United States that are sort of these sweet spots like Napa, for example. And so these are the counties where if population variability is just right, you might see these huge spikes in outbreaks. And so finally, we're going to talk a little bit about our work that we are doing right now. It's brand new work on vine thresholds. So we asked this question of, does local scale movement of lanternfly influence optimal thresholds? Because we know that adults will move in from the surrounding vegetation in Pennsylvania and Virginia and other areas where they've studied this, and will move into these vineyards and then impact the grapevines. And then growers will be monitoring their grapevines, and at some threshold then, they want to initiate some type of control action. And so we developed a 3-stage local scale diffusive movement model. It has 3 stages: a seeking stage, a feeding stage, and a flying stage. And then we've been running experiments to try and parameterize this. So this is an experiment run where we try to parameterize the seeking stage and basically ask how strongly are adults attracted to tall objects like trees, like we see a lot of lanternfly walking up trees, for example. We did some laboratory experiments, and what we found is that, yes, and we're able to now put numbers on it, lanternfly are attracted to these tall objects, and we're able to parameterize the models based on that. And then we're trying to parameterize the feeding stage by asking this question of how long do adults stay on trees? And we're doing this by fingerprinting, where every single spot pattern on the lanternfly is unique, just like your fingerprint. And so we can take multiple pictures of lanternfly on trees, digitize those, put them through a model, and then get estimates of how long they're found on these trees. And this was done just on Temple campus, and we did about 3,000 photos. And what we found in our initial analysis of about 500 photos is that we've identified 54 unique individuals that have stayed on the trees for multiple weeks, and the longest one that we've seen staying on a particular tree is about 3 weeks in time. So now we have an estimate then for that feeding stage, and we have other estimates for the flying stage too. And so our objective here is to minimize the time that spotted lanternfly spends on vines. This is our model here. We have a vineyard, we have a forest of very simplified geometry. The more yellow areas of the higher density, and then they're moving in to that vineyard through time. So then we can run different scenarios where we say, after we see 10 spotted lanternfly per vine, we initiate a control action. And so this is the number of lanternfly that you get when you have no treatment through time. And then this is that where you, as soon as you see 10 per vine, you spray, you cause their population decline, and then it increases again as they're moving back into the vineyard. And you get about a 40% reduction. If you wait until 50, you get about a 60% reduction. If you wait until 100, you're waiting a bit too long, you only get a 23% reduction. And so there's clearly variation on when you should actually choose to do your spray optimization. We then took our model and we adapted it to real geometry in the field. This is a place in Maryland, a vineyard in Maryland, where we have individual tree locations. Then we ran 2 different simulations, one where all the lanternfly were just along this road here, and then one where the lanternfly were spread throughout the landscape. And what we found was that local scale movement of where they're moving into strongly influences the optimal thresholds. So what I have here is then for both this initialization along the road and then uniform in the forest, the threshold is about 75 if you had them all coming in from the road, where it's only 43 if you had them uniform through the forest. So you really have to understand the individual movement patterns of lanternfly. And so I've shown you so far that diffusive spread is slowing, but long distance jump spread is accelerating. And so the question really is how much of this is due to that management action that the USDA and states have taken. I've shown you how there are particular property types that you should be looking for. And so we really have to ask that question of how can management optimally focus on these particular invasion hubs? And then we have to understand climatic variability and how we can best predict and manage these spotted lanternfly outbreaks from year to year. And there are some locations in the United States that are just going to be really prone to these outbreaks, and then some that may not be as prone. And then finally, we have to think about how growers can optimally monitor the movement of spotted lanternfly in their surrounding vegetation to identify sort of their own personalized thresholds as we're developing these thresholds through experimental work that you'll see later on. So thank you, everyone, for this time, and I'm happy to take any questions if there's time.
Thank you, Matt. There's one question in the chat. Somebody's saying, I assume stadiums are somewhat biased due to high number of people, and this also gets at general observation bias. Do you see that in your and factor into your models?
So the null model that we use tries to deal with that observation bias because we thought a lot about that. And then the data that we're using are surveys by state agencies and not necessarily just the population, general population surveying. And so we try to account for it for that way.
Great. The other question here was when you say railroad stations, what category of rail are you looking at?
Yeah, here we looked at, in this particular data set, we used all stations, both passenger and shipping yards as well. And then we're trying to refine that to sort of partition those two out. One way that we partitioned that out was by just looking at the density of rail, and those sort of shipping yards, those had really high density of rail, and that's sort of where we see the most statistical significance.
Fantastic.
Let's see. Donald, yeah, is there still time, Jacob, or should I hop off?
I think, yeah, we should move on to the next speaker. But if you have the time to put answers in the chat, that would be great. Thank you, Matt. It's always great to hear from you. Okay, our next speaker is Holly Shugart from Pennsylvania State University.
So today we're going to talk to you about some work we've been doing in the urban lab at Penn State, and we're going to talk about adult spotted lanternfly weight and how that's correlated with survival trends on certain specialty crop hosts. We'll also talk a little bit about nymphs. There we go. So I'm going to skip a lot of the intro about this insect. We're all here because we recognize this is a serious invasive insect. But I'm going to focus on one aspect of their biology, and that is that they are highly prolificous. So what does that mean? This insect feeds on a lot of different plants at all life stages, particularly as nymphs, but really at all life stages. And we find, our lab and other labs studying this have found that adult survival is variable on different hosts. And so we really wanted to better understand host plant impacts and also the effects on the insect's body condition. So we chose to focus on their weight. So how does their weight change? And we use this as a proxy for body condition. We feel that this provides an additional dimension of the pest-host relationship compared to survival trends alone. We also looked at some nymphal survival and development. And this is kind of with the thinking of, what if an egg mass arrives in a new location? We need to know if those nymphs hatch out, what hosts can they survive on? The longer they survive, it is an increase of the risk of establishment in that new location. And we also need to know if they can survive to be an adult. If they can make it to adult, this incredibly increases the risk of dispersal and the risk of reproduction.
Holly, sorry to interrupt.
Yes.
It dropped out of presenter display. Can you swap the displays?
It looks like it's in presenter display to me. Let me try that again.
There we go.
Okay.
Thank you.
All right. So if you're going to track individual insect weight, the first step is you have to number your individuals. So we used the large size of this insect to our advantage, and we just wrote numbers on their wings. You can see individual 20 and individual 30 here. And we did that for 50 adult females and tracked the individual weight every 2 to 3 days. On 7 specialty crop hosts, we compared this to Ailanthus or tree of heaven. We also tracked their survival, so we knew when each individual died, and we only took live weights. So for nymphs, we collected early first instar nymphs, caged them to the host, tracked survival and host impacts, and we also tracked their developmental milestones. Okay, so let's look at what plants we worked with for the adult work here. So 2 species of grapevine. This is Vitis vinifero, Sauvignon Blanc, that's a wine grape. We also did muscadine grape, that's Black Beauty variety. We looked at fuzzy kiwi, and it was Hayward variety. We looked at 2 varieties of hops, Cascade and Centennial. We looked at citrus, and we used variety Valencia. We looked at avocado. Haas variety, and we compared all of this to Tree of Heaven. So again, 50 females tracked per host. We started the experiment with unmated pre-reproductive females. These are forced feeding assays. We didn't give them a choice about what host they were on. They were forced to feed on a specific host. We looked at percent survival at each time point and total days survived. And we did a regression analysis to compare the link between weight gained or weight lost and survival duration. And so this is what that looks like. So we've got the dark green here is avocado, day 3. So these are weights on the vertical axis, and along the bottom, these are the days that they were weighed out to the last day they survived. And each individual color, like the dark green here is avocado, they died at day 3, each color the different host plants. This sort of orange peach color represents citrus. They all died by day 5. The 2 green here, the 2 lighter green and the pink, the 2 lighter green are hops, and the pink one is muscadine grapes. They all died by day 8. And we had 3 hosts where they lasted quite a lot longer. Fuzzy kiwi, they were, last day they were counted was day 26. Last day they were found alive on Ailanthus was day 33. And with Sauvignon Blanc grape, they were alive until day 40. So let's look more specifically at their weights. And I've added it's basically the same data we just looked at, but in a table format. I added percent weight change here because I think that's a little easier to understand. And I reoriented things. So we've got the category where they gained the most weight here at the top, all the way down to where they lost the most weight at the bottom. And so the 2 blue here, these are hosts where they gained weight. So on the Sauvignon Blanc grapevine, they gained 13% of their body mass. On Ailanthus, they gained 7%. Muscadine grape was interesting. It was exactly the same number, out to 4 decimal points, so no change. And on all other hosts, they lost weight. Fuzzy kiwi, where they survived for 26 days, they only lost 2.5%, but in everything else, it was quite a lot more. Hops, 14 and 16% lost. Avocado, 18%, and 41%, though, for Valencia citrus, which is really interesting. So we wanted to show kind of more specifically the changes in weight and survival trends, and sort of got kind of 0 weight change. These lost, these gained, and we feel that 82% of the weight changes can be explained by host species, which is really, really interesting. Okay, so hops, muscadine grapes, citrus, avocado, less suitable for adults. Put a pin in that. And we think that fuzzy kiwi may support adult populations longer term. Adults survive for 26 days, even though they lost 2.5% of their body mass. Grapevine and Ailanthus are the most suitable hosts. Adults survive longer and gain weight compared to other hosts. Remember, plus 13% for Sauvignon Blanc and plus 7% for Ailanthus. Now let's look at nymphal survival and development. And we focused this work with citrus and avocado. So we used 5 to 6 foot citrus plants. Again, this is Valencia variety, and 6 to 7 foot avocado trees, and this was Haas variety. We field collected early first instars and added them to the trees. We constrained them by the mesh bags, as you can see here in the picture, 50 nymphs per. We had 8 trees for each host that received nymphs and 4 control plants. And then we also tracked developmental milestones and compared them to Ailanthus. So let's look at survival first. Nymphs survived for 13 days on avocado. They only made it to second instar. But nymphs survived for 95 days on citrus. We did not expect that. So let's break that down a little bit more and compare developmental milestones. So this chart, these numbers are days from the beginning of the experiment where each of these instars appeared. So this row here, 8 days after the beginning of the experiment, we saw our first second instars for each, 18 and 21% until we saw the first third instars and so on. So starting out, citrus development on citrus is slightly faster compared to Ailanthus, up to fourth instar. And then development slows during the fourth instar. We had fourth instars on citrus from day 30 to day 95, or for a total of 65 days. And then on day 68, we did have one adult emerge. And that's the first time that has been documented. Now, when we compare fourth instars on Ailanthus to citrus, we had fourth instars appear in citrus at day 30 and on Ailanthus at day 36. But then we started seeing on Ailanthus, they became adult starting at day 45. And within 10 days or so, 95% of the insects on Ailanthus had become adult. But on citrus, they were just fourth instars until the last one died on day 95. So let's talk about what it looked like to have the insects on citrus for so long. So the nymphs feeding on citrus produced copious quantities of sticky honeydew. It was not the most fun thing I've ever done to dig around and count nymphs in these very sticky trees and sticky bags at some point. And we worried that the honeydew could be a substrate for fungal pathogen growth. This could be a problem. For our purposes in maintaining these plants in a greenhouse setting, we just rinsed the plants off because we wanted to keep them and track how they did following this feeding exposure. Once we remove the honeydew, the test plants are doing great. So good news, bad news, right? So let's talk more about that. So with spotted lanternfly and citrus, what are the concerns? The concerns are that they survive for 95 days as nymphs, that they can complete their life cycle on citrus. We had that one male adult emerge. It's also a concern that the nymphs produce so much sticky honeydew, and this could be a problem for fungal pathogens for growers and groves. But there's good news too. So the adults did not survive well on citrus. They only survived for 5 days, and they lost 41% of their body mass, which is significant. But another piece of good news is the plants were not seriously damaged by spotted lanternfly feeding. Even after 95 days of nymphal feeding, those plants are still alive and doing well. Okay, data summary. So grapevine is most at risk from adults. We do worry that other hosts like citrus and hops might support populations for short periods of time. And the real worry is where there's the coexistence of these hosts with grapevine. Fuzzy kiwi may support adult populations longer term. They survive for 26 days, much longer than on other hosts. And we've shown that spotted lanternfly adult weight is linked to survival duration. And this can be a useful measure for host suitability and for insect condition. And it's really logical, right? Weight is lost on less suitable hosts. Weight is gained on more suitable hosts. And we found that nymphs survive well on citrus. They survive for 95 days and they can survive to adulthood. They also produce a lot of honeydew while they're feeding. We worry about all of this together as a problem for these areas where grapevine and citrus coexist. Of course, we're thinking of places like California. So conclusions and broader implications. Pest-host interactions are really complex in this highly prolificous insect. They can feed on so many things, and we get pieces of the puzzle every time we do studies like this. For our purposes in a greenhouse, the insects responded really differently to the different hosts. But also importantly, these differences are highly dependent on life stage. Citrus is a great example. The adults lost 41% of their body mass and died within 5 days, but the nymphs survived for 95 days. More broadly, these interactions are going to depend on when they arrive in a new location, host availability, how that intersects with host preference, climate, which hosts are available when each life stage needs them. It's really complicated to figure all of this out. And that's all I have. I'd like to thank everyone who's contributed in some way to this research and our funders, and I'm happy to answer any questions.
Thank you, Holly. Just one question here, asking if there are any other crops that are on the list to come.
I mean, there are studies that have been working on a lot of different crops. I don't have them memorized, so I'm sorry, I don't really have a good answer for that.
Okay. There's another part of this question. If SLF fed on both grapes and tree of heaven, how would that impact the weights?
The weights specifically have not been studied in other studies, but there are some folks who have, given insects a mixed diet. Melody Keena said that earlier, that some of her work had tree of heaven and concord, right? But they weren't necessarily weighing their insects in the same way that we were. And so we each sort of have pieces of that puzzle, but it's not a yes or no answer. Likely, they would do better. That seems to be kind of a theme in these plant feeding insects. You give them a better variety, especially this really highly polyphagous insect. The more variety they have, the better they do.
Absolutely. Yeah, I do want to remind folks that the website stopslf.org, which is maintained by the Northeastern IPM Center, does aggregate all of the published research and has for several years now. It's a tremendous resource. And I do believe there's various work there that goes into weight, survival, and maturity based on access to multiple food sources. And I believe your work is also there, Holly. Is that right?
Yes.
All right. Well, thank you again, Holly. And we are going to move right along. Our next speaker is Hannah Broadley. Hannah is coming to us from USDA APHIS, where they are vetting various parasitoids for the classic biocontrol of spotted lanternfly. Hannah, if you're ready.
Yes, can you hear me?
Yes, the volume's a little bit low.
That is unfortunately a chronic problem with my current microphone, so I
yeah.
apologize for that. People have told me it will come in, it starts quiet and comes in a little bit louder.
Okay, we'll hope for that.
Sound okay?
Yep, we can hear you.
Okay, all right. Hopefully I get the correct screen. Share. Share that screen. Did that come in for you?
Yes, we have the view.
Great. Okay. Let me make sure I can advance through them. Yeah, so as you said in my intro, thank you. I'm a scientist with the Forest Pest Methods Lab. We're a lab of USDA APHIS, Plant Protection and Quarantine, and we've been working on developing classical biocontrol for spotted lanternfly. Our focus right now is on Dryinus sinicus. So just a reminder what biocontrol is. So biocontrol is the reduction or control of a pest by an introduced natural enemy. This typically involves an active human role. And the natural enemy that we are focusing on for our work is a parasitoid, and a parasitoid is an insect whose larvae lives as a parasite, typically in other insects, and eventually kills their host. And this work is focusing on classical biocontrol, which is the importation of a natural enemy from a foreign country for sustained control of a pest of foreign origin. And successful biocontrol can provide enduring pest control. These biocontrol agents can reproduce and disperse without further human assistance after they're introduced, and they will persist after the pest population is reduced to low density. So upfront, it takes a lot of work to develop these techniques, but when they are successful and able to be released, they're very cost effective. Classical biocontrol is best pursued when a species is not native, so that applies for spotted lanternfly here. when a species has established for at least 5 years. So it takes a long time to develop biocontrol. So we want to know that this is a species that's not going to pop up as a pest and then disappear and not be a pest anymore. So spotted lanternfly has been an enduring pest for more than that amount of time. It's causing economic and ecological damage. Again, there's a lot of effort to develop biocontrol, so it's necessary to know that the development of it will be worth the cost of the pest, outweigh the cost of the pest. And we need to know that eradication and control by other means alone is not possible. So the idea is that this can layer in as a technique to use to control spotted lanternfly. There's a lot of steps for developing biocontrol. So here for spotted lanternfly, we first need to evaluate the spread of spotted lanternfly and the role of native and resident parasitoids and other natural enemies in the US. Investigation of the insect and its native range. So this is in China. So that's foreign exploration to see what natural enemies exist in its native range that are holding the populations down. Select candidate agents, then import and develop laboratory colonies of these insects, so develop rearing of them. Complete host range testing and determine efficacy of the candidate biocontrol agents, and then apply for and receive permits for release of these candidate agents. And then last step is to be able to release those agents and complete post-release evaluations. I want to right here acknowledge the many different collaborators that we've been working on for the developing classical biocontrol. So we have our local team members here. So in blue listed here are our federal and state collaborators. And then green are university collaborators for this work. And then to complete this work, we also have numerous international collaborators. So right from the forefront, I want to acknowledge all of those collaborators. Okay, so we went through this list of different steps to complete for developing classical biocontrol. I'm going to quickly go through each of these pieces and give you a short update. Each of these could be its own talk in itself, but a short update of where the work is at this time with those. So native and resident parasitoids. There are some native and resident parasitoids that have been detected attacking spotted lanternfly. So Oenceradus, this is a parasitoid primarily of spongy moth, and Anastatus reduvii. These are two that our lab teams, as well as others, have done some research on. So they are out there. However, these are not causing population control at all. So for Anastatus reduvii, for example, we're seeing for our surveys across the range of spotted lanternfly, less than 1% of egg masses are parasitized by this species, and then less than 0.05% of individual eggs. I don't have the numbers right off the top of my head for Oenceradus, but I know that those levels are very low as well. These, unfortunately, are not causing any population level control. Foreign exploration. So we've been working with collaborators out of the Chinese Academy of Forestry in Beijing on spotted lanternfly and looking for parasitoids since 2015. And doing extensive surveys across spotted lanternfly's native range to determine what natural enemies are there, and looking at the populations across China where typically spotted lanternfly is not a pest species. Out of this work, these two parasitoids have come out as important and prominent parasitoids. So here we are selecting candidate agents. These two parasitoids, our initial work has been on Anastatus orientalis. So folks that might have heard talks previously by me or collaborators on developing classical biocontrol for spotted lanternfly, likely heard more about Anastatus orientalis. This is an egg parasitoid, and in China has a pretty high level of parasitism, with 2 generations per year and 30 to 40% parasitism. It also was selected as a biocontrol agent for classical biocontrol for invasive spotted lanternfly in South Korea. So this was the species that we focused on initially. However, after extensive work on multiple haplotypes of this species, we have not found it to be specific enough, host specific enough to consider further. So we are not working on this species anymore. It's unfortunate because it was used previously successfully in South Korea. It's easy to rear, high parasitism, but it's not host specific enough to work with. So we are now focused on Dryinus sinicus. This is our nymphal parasitoid. It's a also a really neat insect. It's a Dryinid, so family Dryinid. It's native to China. It attacks second and third instar nymphs with 1 generation per year. It's got these really amazing front forelegs, these raptorial forelegs that it will reach out and grasp its prey with. So in that photo there on the left, you can see them sort of held up in front, sort of almost like a praying mantis holding those forelegs towards its body. And then the parasitoid develops out of the SLF nymph in a protective sac called the thylacine. Here is a beautiful picture that was taken by one of the technicians, lead technicians in the lab. Corinne took this awesome picture of one of our wasps holding on to grasping a nymph and host feeding on it and parasitizing it. And then also here I'm showing the life cycle with Dryinid being out in the field, mainly end of May, June, during that time for parasitism and attack. Then it spends the rest of the summer developing and spinning its cocoons, overwintering as cocoons, and then the life cycle repeats from there. Okay, so back to this. So we've selected our candidate agents right now. That's the nymphal parasitoid, Dryinus sinicus. And then we have these now imported and we have lab colonies of these. So to maintain these colonies, we have plants, we maintain colonies of nymphs, and then we have the wasps that feed on those nymphs and parasitize those nymphs. We have been able to manipulate the life cycle so that we can get more than 1 generation per year. That helps speed along the work and helps us produce more cocoons. Here is showing in 2023 and then last year what our cocoon production was. So last year we ran into some issues with cocoons going into a diapause that we couldn't break. So we had slightly lower numbers of cocoons produced last year, but we're still up into the thousands and still have a lot to work with. We also have a secondary colony at one of the ARS facilities because it's really hard to get these samples out of China at this point. So we have a strong colony producing over 1,000 cocoons per year. Sorry, I went backwards. Okay, so then we're on to step 5 of this process, completing host range testing and looking at the efficacy of the species. We start our host range testing by completing no choice testing. So that means that we give those wasps either a non-target species or a control, which is spotted lanternfly nymph, and we don't give them a choice between those two. And we do this because it's a more conservative test to start with. If we do see any parasitism or attack with these no choice tests, then we move on to a choice test, which I'll show you next. We are also completing these over doing them sequentially. So we give each wasp a spotted lanternfly nymph to feed on to start with. Then we give them a non-target for the first day for an hour observational period. we let them rest, then we give them a target, so a spotted lanternfly nymph, non-target, target, non-target. And so the reason we do that is so for each wasp, we get multiple information on its attack over a whole week. And we see if there's any, you know, as it goes towards the end of the week, if it's particularly hungry, is it more likely to attack? Or if it's been exposed to a non-target, is it more likely to be interested in that non-target later? So this gives a lot more information doing it sequentially. In our first year of testing, these were the species that we were able to test. So we have it listed here by group names with the out groups that we're calling kind of at the top there. And then we go down to the plant hoppers on the bottom of this table here and the fulgurids at the very bottom. And what we found here is that in all of those that we test in this first year in 2023, we found no attack and no interest in these non-targets. So a really great start. Okay. Then last year, we were able to add more species and we were able to get more of these plant hoppers into testing. It took us sort of a pilot year to figure out how to find these plant hoppers, how to rear them. Generally, a lot of our plant hoppers are understudied and really challenging to find and rear. So it took that pilot year to get these. But we've now added more of these plant hopper groups and added more closely related species to spotted lanternfly. So added 10 additional species. What we did find with these plant hoppers, that we did see low rates of host feeding with these species. However, we did not see any parasitism at all with them. And again, these are in no choice tests, so very conservative tests. Here's showing a chart of what that looks like. So blue here is showing the attack rate on the non-target, and orange here showing the attack rate on the controls, so the spotted lanternfly nymphs. And what you can see here across the board is, yes, they will attack these species and try to host feed on them, but they're much less interested in attacking these non-targets than they are spotted lanternfly nymphs. Also, this work is still, we don't have our full replicates of this, but again, why it's unfortunate, there is some attack with no parasitism, and with this attack being lower than the targets, this looks pretty good. We're working with collaborators in China to do host range testing as well, and they've found same thing, that just the fulgurid, or similar, they found attack on their fulgurid that they tested, but no parasitism and no attack on any other species that they looked at, and no attack on any of the other plant hoppers either. So for these species that we did see host feeding on, we are moving on to choice testing. So we are also running this over a period of a week, doing sequential testing. But we do the non-target and the target together so that the wasp has a choice between two options of which it prefers. These are also preliminary. We don't have the full data set for these yet, but we are seeing a very clear pattern of reduction of attack. So very strong choices for those wasps when they have an option of a spotted lanternfly nymph or one of these non-target plant hoppers. They are mostly choosing the spotted lanternfly nymph over these. Again, we do see a little bit of attack, no parasitism, but that attack rate is much, much lowered. So again, while it's unfortunate that there is attack, this is actually really good results, and we're happy with this. So, here we are. We've gone through with Dryinus sinicus, we've gone through those first 5 steps. We are in constant communication with the permitting team to make sure we're on track. And they also feel that we're on track with the species. So if this season goes well, we are planning to apply for and put in a permit for release in the fall. So we have started to draft up that permit. We still need more testing this field season, but we've started to draft up that permit with the hope of getting it in the fall. So overall, we've made really significant progress on the host range testing of Dryinus sinicus. We are not seeing any parasitism. We do see low rates of host feeding, but nothing that is it's a concern, but not, we don't need to stop our testing with this. We'll need to get more data, but it all looks very promising. We have, particularly with those choice testing, we're seeing a strong choice for spotted lanternfly over any of these non-targets. We have additional taxonomic and geographic breadth of non-targets that we're getting into testing this year. And we're aiming to have sufficient data to be able to put into a permit application at the end of this field season. So with that, thank you very much. I really appreciate being able to share these updates on Dryinus sinicus, which is just a really, really cool parasitoid. And if there's any extra time, I will take any questions.
Thank you, Hannah. Very exciting to see how close we're coming. There's one question in the chat here from Scott Salom. How does the euploid function with 2 generations when the lanternfly only has 1 generation per year?
How does Dryinus function with two generations? Oh no, the euploid. That's what you said.
Yes.
Yeah, so Anastatis orientalis, the egg parasitoid, what it would do, or what it does in the field in China, is it uses the eggs. So it's able to use the eggs when they're laid fresh in the fall, and then it emerges in the spring and parasitizes those again, in theory. However, one of our big concerns about that species is we see a lot of non-target attack. So we do see it reusing those eggs again in the spring, but we also were seeing generations that emerged after the SLF nymphs, meaning that it would need to use a non-target species, and we would see generations emerging in the summer. So in theory, it can survive with spotted lanternfly, but in practice, we do not see that happening. So that's another reason that it was pretty clear we should not pursue that species any further.
All right. There's quite a few questions in the Q&A, and I'm going to ask you if you have time to type those answers.
I'd be happy to.
And thank you again.
All right, and now, because I have the other screen up, now my screen for sharing, I'm trying to see if I can unshare. Okay.
All right.
Got it. All right, thank you very much.
Thank you. Our next speaker here will be Flor Acevedo. Flor is from Penn State and is working on quantifying the effects of lanternflies on grapevines. Flor?
Thank you. My screen looks a little weird, doesn't it? I try to share again. Looks very strange. Are you seeing the presentation?
We are in presenter mode now, so you should be able to switch.
Like this? Is that better?
Yes.
Okay, fantastic. All right. So today I'll be talking about the effects of spotted lanternfly feeding on grapevine yield and juice and wine chemical parameters. First of all, I would like to acknowledge the people that have been working on this project. This has been a joint effort among 4 different labs. We have Michaela's lab that focuses on grapevine physiology. Misha's lab that focuses on food sciences, Claudia's lab in agricultural economics, and my lab that focuses on entomology. And we also have, of course, postdocs, students, and technicians that have been doing the work. So spotted lanternfly has been recognized as a pest in vineyards in the US. And the insect, we know the insect affects grapevine health, affects yield and fruit quality, and therefore it must be controlled. All insect stages, eggs, nymphs, and adults can be found in vineyards. But the adults are the ones that are more problematic because they are more mobile and also because they extract large amounts of plant sap during feeding. So this graph shows how spotted lanternfly populations fluctuate in vineyards. The data came from a 3-year study that was carried out by Hetrich in 8 different vineyards infested with lanternfly in Pennsylvania. And the graph shows the number of lanternfly individuals in vines from May to November. And we can see that the nymphs can be found in the vineyards, especially early in the season. But the greatest numbers of lanternfly individuals are seen at the end of the growing season. And these are adults. Adults that feed voraciously on grape and must be controlled to avoid damage. This peak of lanternfly infestation overlaps with harvest, which is problematic for management. So this information has been super useful for us to determine where the management efforts should be focused. Another piece of information that is relevant for management is the number of insects per vine that would decrease yield and reduce juice and wine quality. So to test that, we selected 2 widely grown Vitis vinifera cultivars, Chardonnay and Cabernet Franc. And these 2 cultivars were selected because they differ in a few aspects. Chardonnay is a white cultivar that buds early and ripens early, and it also gets harvested early. So in Pennsylvania, this cultivar gets harvested from late August to early September, and depending on weather conditions. And Cabernet Franc is a red cultivar that has a later bud break. It ripens later in the season, and it usually gets harvested by the end of September in Pennsylvania. So it is important that they have different ripening times because they will be exposed to lanternfly adult feeding different times also, and that might have an effect in the fruit. Okay, so to investigate the relationship of spotted lanternfly feeding on grapevine yield and juice and wine chemistry, we used 20-year-old grapevines that were not previously exposed to lanternfly. We built large enclosures and infested these grapevines with different spotted lanternfly numbers. Those numbers range from 0, 1, 2, 3, all the way to 15 insects per shoot. This translates to about 20, 23 to 300 insects per plant, a little higher in other cases, and it did vary per year. And we infested these grapevines with lanternflies during 3 consecutive years. So each vine was infested with the same number of insects throughout the experiment. And at the end of the season, we harvested the vines and collected fruit samples for analysis. In our experiments, we use nymphs and we use adults separately. So the nymphs were placed on the vines from early June to mid-August. and the adults were introduced in mid-August and they were killed in mid-October. Both nymphs and adults fed on the vines for about 2 months, 50, 60, 70 days, so about 2 months. So the big difference here between the two cultivars was the time that adults fed on those grapevines before harvest. In Chardonnay, it was about 30 days, and in Cabernet Franc, it ranged from about 45 to 50 days each year. And then we tested the effects of the nymphs for only 2 years, whereas the effect of the adults was tested for 3 years. And we quantify yield and standardize the total weight of the fruit by the number of shoots per vine. In the juice, we measure pH, degrees Brix, and titratable acidity. And we made wine also with those grapes, and then in that wine, we quantify phenolics and tannins. Okay, so these are our results. I'll be presenting the results for the adults, and then at the end, I'll briefly summarize the results for the nymphs. So these graphs show the relationship between yield in the y-axis and spider lanternfly infestation, by different numbers in the x-axis. Yield, again, was standardized by the number of shoots per vine. And what we see here is that the association between yield and infestation with different spider lanternfly numbers was not significant, as you can see by the p-values that I'll be presented, that are here, and I will be presenting those throughout the slides. So there was no significant association between these two variables. And that was during the first year of the study for the two cultivars. But in the second year of consecutive insect infestation, we did see a decrease in yield in vines infested with adults for both cultivars. And the response was stronger for Cabernet Franc. As you can see, the line is steeper, and also the R-square is higher. And in the third year, we found a sharper decrease in yield with higher lanternfly densities. This response was again stronger in Cabernet Franc, but significant in both cultivars. Okay, juice pH. In the first year of insect infestation, there was no significant association between juice pH and spider lanternfly density in Chardonnay, but there was a decrease in pH in Cabernet Franc. In the second year, there was no significant relationship between pH and lanternfly density in either cultivar. And same for the third year, in which the results were not significant. Okay, for total soluble sugars in grape juice, we found a marginally significant decrease in Chardonnay. As you can see, the p-value was 0.056. And there was a significant decrease in sugars in Cabernet Franc during the first year of lanternfly infestation. In the second year, we found a significant decrease in sugar levels, again in Cabernet Franc, but not in Chardonnay. And in the third year, we found similar results, a decrease in sugars in Cabernet Franc, but not in Chardonnay. For titratable acidity, we found in grape juice, we found no significant association between the TA levels in the juice and spider lanternfly density in year 1 for either cultivar. Same in year 2, there was no significant results. But in year 3, there was a significant increase in TA levels as the insect densities increase in Cabernet Franc only. Okay, for phenolics in the wine, we found a significant decrease in the concentration of phenolics in year 1, but not in year 2. And this was only done in the red cultivar in Cabernet Franc. We have not done this analysis in Chardonnay yet. So again, significant in year 1, but not in year 2. Similar for tannins, there was a significant decrease in tannins in year 1, but not in year 2 for Cabernet Franc. The samples for year 3, we are still in the process of analyzing them. Okay, so this is a summary of what we have found so far. Our results indicate a consistent decrease in yield for the 2 cultivars during the 2nd and 3rd year of spotted lanternfly infestation. There was also a consistent decrease in sugars for the red cultivar in the 3 years that the experiment was conducted. And in some years, as you can see, we found a decrease in pH, higher TA in some of them, lower phenolics and tannins, but there hasn't been consistent through the years. Okay, how about the NIMS? For the NIMS, most results were not significant, except for lower sugar levels in Cabernet Franc use during year 1. So, in summary, spotted lanternfly feeding by adults was associated with a reduction in yield in the 2nd and 3rd year after consecutive infestation. Feeding by these adults was associated with a decrease in sugars, pH, phenolics, and tannins within a single season of infestation for Cabernet Franc, but not for Chardonnay. So during the first year that lanternflies fed on these vines, no effect in yield, but there was an effect in juice chemistry and wine chemistry. For the 2 cultivars tested, we found a stronger response in the red cultivar, as you saw. And for NIMS don't appear to be as damaging as adults, and there are experimental conditions, of course. Grapevines were able to withstand 2 months of constant feeding by up to 300 NIMS without showing signs or consistent signs of damage or effects on juice and wine chemistry. So this is actually good news. Okay, our study, however, has some limitations, of course, and the results of this study represent extreme circumstances in which a spotted lanternfly was forced to feed on the same vines for an extended period of time, approximately 60 to 70 days without management. And the grapevines we had available for this study were a little old. It is possible that younger vines might have a different response. We don't know that. But still, the study is useful to understand the relationship of different insect densities on yield and juice quality under extreme conditions. However, further research is needed to determine how long grapevines can tolerate insect feeding without experiencing negative effects in either plant health or juice or wine chemical characteristics. And lastly, I would like to thank the funding agencies that have made this research possible, including the Pennsylvania Department of Agriculture, USDA NIFA, the Pennsylvania Wine Marketing and Research Board, the New York Wine and Grape Foundation, and of course, the Penn State College of Ag. And thank you for your attention. I can answer maybe a question if you have it, and if not, if we don't have time, put it in the Q&A or I'll answer. Thank you.
Thank you, Flor. All right. If Scott Salem, are you ready to present? If so, we can just swap our order here.
Yeah, I'm able to present. Yes.
Excellent. All right, so we're going to diverge from the schedule just a little bit. Scott is going to give us an update on biocontrol coming to us from Virginia Tech. And after that, we will go back and see if Zach is ready. So, Scott Salem, your show.
Okay, how's this showing up to you guys?
Looks good.
Okay, great. Hello, everybody. Pleasure to be here talking with you this. I think I was a last minute addition, thanks to Julie Urban, since we have a little bit of an update regarding a biocontrol agent or prospective biocontrol agent for Tree of Heaven that we've been working on for close to 2 decades. And so in recent years, we haven't talked a lot about it because it has not only been under quarantine, but it has been going, we've been pushing to petition to have permission to release it. And so there haven't been a lot of opportunities to talk about this to everybody in the community that's interested in trying to manage Tree of Heaven for the purposes of impacting spotted lanternfly population. So this is a little bit of a coming out story for those of you that may not be familiar with this weevil. So I'm going to pretty much overview what work's been done and then talk very briefly about, where we're at right now. All right, so I want to advance this. Perfect. So let's see if I'm going to get the laser pointer here. Tree of Heaven, this is our friend. We know all about it. You've seen many pictures like this. Pretty ubiquitous in our environment in the East and introduced from China, growing in disturbed sites. Trying to get rid of it. You can see in the images here that you cut them down and they just come back stronger. Hundreds of thousands of seeds that are produced in mature trees. So you have sexual reproduction as well as vegetative reproduction, and the fact that they sprout from roots and stumps, grow in every different condition that you can imagine. So that's our target. And then there's this insect that we found in China, or we visited our colleagues in China and learned about this insect, which is an important pest of Tree of Heaven there, and is also considered or known to be host specific there. And so this weevil, we affectionately called it the panda weevil, but we've had to propose, or recently decided to propose, a common name for this with the NSOC of America, which was approved by the, we just learned last week, approved by the Common Names Committee at this point, and are proposing the name Ailanthus trunk weevil. And as adults, these insects will feed on the foliage and stems of the tree and the canopy, and it helps them become reproductively mature. Once they are, then they will oviposit in the stems and the trunks of the trees, eggs going into the bark. Larvae develop in cambial tissue, pupating, and then the insects pupating, basically in the outer sapwood of the tree. And generally one generation per year that we're aware of. So that's just a very quick overview of its life cycle. I'll get more into the insect and its impact in China. So here's a map that shows you the distribution of both Ailanthus and the Ailanthus trunk weevil in China. And you can see it's a perfect overlap. And the red dots represent areas where the insect's been collected and has been shipped to us at Virginia Tech, where we've been studying it under quarantine for close to 20 years. And this is a Forest Service map. It just shows you the distribution of Ailanthus in the eastern US. So I like this better than the county maps, which are not quite as accurate. So you can see that this Interstate 81 corridor is packed with Ailanthus, right? Okay, so in China, Tree of Heaven is really a desirable tree. They grow not only in urban environments as a shade tree, but they're also grown for, fiber, And generally, this is often done, you know, in areas that aren't ideal for its growth. And so when this tree is attacked, this weevil comes in high numbers and can kill trees. And so this just shows you some of the damage that you can see on these trees, the emergence, the galleries underneath in the outer sapwood, and you know the insect in that outer sapwood. And so they're managed for. There's insecticide treatments of these trees when they're under attack, and so it's considered a big problem in China. You know, but as much as we studied this insect, we realized that we weren't able to find this insect like actively attacking healthy trees. And so we combined our efforts with our Chinese colleagues and conducted a study that demonstrated that the greater stress that you put the tree under, and that usually meant pulling bark away and girdling it, the more colonization of the tree occurred with these weevils. So healthy trees, not much attacks. But the more girdling that took place, the greater the number of attacks that took place. So this will have implications in terms of how we approach using this insect if we get a chance to in the future. And there are options in which we can stress trees and then introduce these insects down the road. So obviously, how are we going to get approval for this insect? After beginning to study it in quarantine, we did basically host range testing on 29 non-targets. These had to be approved by the technical advisory group, administered through USDA APHIS. And in this two-choice test, it shows you in foliage feeding that there was tree of heaven, and only corkwood and lemon showed any kind of real activity. Corkwood is a rare species related to tree of heaven that occurs in several states in the southern US. There are 2 species of corkwood. And as a result, this has slowed the approval process up. But nevertheless, you can see the tree heaven is highly preferred. In terms of no choice testing, you can see how well the insects survive feeding on the different host species, and you can see tree heaven is far superior to anything else. Looking at larval development in no choice testing, you can see that Ailanthus, tree of heaven, better than anything else, but corkwood, which is Latinaria, shows that there is some ability to develop and also successfully develop in that host. So again, we went through multiple efforts, multiple iterations of testing until things went well for us. In the lab, how can we rear these? So we developed a rearing procedure that allowed the insects in these cages in quarantine to feed and oviposit for a couple of weeks. You can notice that this bolt is sitting in water, which would cause these bolts to sprout and maintain integrity for larval development. And then we would take these bolts out and we'd sit them in these racks, also containing water. And then we'd cage them up with this mesh tent-like material. And months to up to a year later, insects would be emerging out of these bolts. So fairly labor intensive, stinky, smelly, and all that kind of stuff, but very effective. And we've had multiple introductions from China to infuse the colony with new insects. The last trip taking place in 2019. So now I want to switch a little bit. You've probably heard a lot over the years. Many of you have been pretty interested in this naturally occurring fungus, Verticillium non-alfalfae, which was first found in Pennsylvania. We found it in Virginia. Then it was also found in Ohio. And we're currently working feverishly towards getting this approved and registered through EPA. So this is going to be a potential bioherbicide that we can apply on the landscape, and is part of the area-wide USDA ARS project that is ongoing and led by Tracy Lesky. Some of you may have heard of this. Anyway, I'm bringing this up because this is a pathogen that can spread through the roots of trees once the trees are inoculated, and can spread on its own in a stand, and is fairly quick at killing the trees, and is very specific in attacking tree of heaven. So This is a native pathogen that has been found and has considerable potential. And we knew this way back when we were working with this weevil in the quarantine. And so we wanted to see if this weevil could actually transport the fungus to uninfected trees. And so we ran tests where we looked at weevils that were walking over the pathogen on these agar plates. And then we would also take those weevils and we'd either have them feed on uninfected plants and infected plants. Those were the two treatments. Then we'd release them and cage them on uninfected plants. And then after 72 hours, cut those twigs, give them 3 weeks, and then put them on these agar plates and see if the fungus was being transmitted. So again, this is very quick, very much of an overview. This was part of a master's student's thesis. And so just the takeaway message here is that if there was tarsal contact on those Petri dishes with the fungus, but they were feeding on non-infected plants, they were able 50% of the time to transmit the fungus to clean seedlings. If they combine the tarsal contact with feeding on infected plants, then that increased up to 75%. So this is an indication, at least in the laboratory, that these insects in the field, once they come in contact with verticillium, that they might be able to transmit the fungus to trees and help spread the fungus along beyond what the fungus itself can do, which tends to be more localized within the stand. So that gives you a very quick overview of where we're at in terms of some, this is what we knew about this insect based on all the research that we had done. So in 2022, we had petitioned in 2021 the USDA Technical Advisory Group for the biocontrol agents of weeds. And in 2022, it was approved. The petition was approved, and it was recommended that brandti be released in North America due to the low risk of direct and indirect non-target impacts on federally listed species or to U.S. agriculture interests. So I'm generally just reading this, but following the writing of a biological assessment by APHIS, this went to the U.S. Fish and Wildlife Service, and a letter of concurrence is pending at the time of this writing. So I learned in early December that this was ready to get approved, and then it's been sitting on the desk, and then, politics has come into play, and we don't know what's going on, but it's still at U.S. Fish and Wildlife Service. So we're expecting that it can continue the process, the petitioning process, and if so, felt this was the time to get out and let everybody know this. We have the weevil, and all we need to do is start getting organized and planning for mass rearing of the insects so that we can really ramp this up. Obviously, in the field, test for releasing, establishing, and then also the potential impacts that this biocontrol agent would have, and try to find some funding to support this work. And it's believed that this would dovetail really nicely into the current USDA ARS area-wide project on spotted lanternfly, which is focusing on biologically based tactics. And this wasn't initially included in this project because we didn't know where it stood with regards to petitioning, but we think we're close. This has been funded from the U.S. Forest Service by the U.S. Forest Service Forest Health Protection for over 15 years. That along with the verticillium work, and then more recently through APHIS, the verticillium work through APHIS to get this approved through EPA. So that's the quick story. I'm sure there are a lot of questions out there for everybody. And so with that, I'm going to stop sharing and see if there's time for questions.
Thank you, Scott. Looks very promising. We are going to jump ahead. There may be some questions in chat, but
Okay.
It sounds so. Thank you very much.
You're welcome.
It sounds like Zach is back online. Zach, how's that looking?
Can you hear me, Jacob?
We can hear you.
So hello, everyone. My name is Zach Hetrich, and I am with the Pennsylvania Department of Agriculture. I am the supervisor for the Western Zone of the Spotted Lanternfly program for PDA. I will show you here in a minute where those zones are, but today, instead of just the West, I'm going to talk about the entire program and give everyone an update on our raw data for the previous year, and then also give you kind of a background on why we do what we do. So we'll move along here. All right, so here is the overview of where our offices are located in Pennsylvania. So my office is out there in the western part in Greensburg, PA, and then we also have an office, our central located office in Harrisburg, and then an eastern office in Palm. So as far as our staffing goes, we have one entomology program manager that manages the entire statewide program. That position is currently vacant, but is being filled shortly. We have 3 plant inspection program specialists, I being one, myself, and then 2 others, one in central and one in the east. And then we have a what we call a treatment crew and a survey crew that are in each zone. So the treatment crew consists of 9 foresters and 3 forest technicians. So that's 3 foresters per zone and 1 forest technician per zone. And then we have a survey crew, which right now we have 2 forest insect pest aid 1s, or we call them FIPAs, which are the main on the ground surveyors. And then we have a forest insect pest aid 2, which kind of is the team lead for the FIPAs. So we're going to talk about what the survey crew does first, and then give you a little bit of information on our data from last year. So the survey crew mostly does visual surveys, trapping, and public reports. So we'll focus on visual surveys first. So some strategies we use for visual surveys. We focus on high-risk pathways, vectors for long-distance dispersal in areas with high volume storage, and especially disturbed sites as well. And we use a little bit of aerial imaging, local knowledge, and other prediction models to try to figure out where these sites are within Pennsylvania to look. And so now this is the numbers for last year for our visual surveys for the entire state. We had a total of 31,882 surveys across the state, and 5,683 of those being positive and 26,000 being negative. And then we also had a few undetermined, but those are, we try not to do those as much, but those are more like maybe an egg mass or something that can't be completely identified, but we try not to use undetermined that much in our surveys. All right, so I want to talk a little bit about Erie. So Erie is one of our highest priority areas. It is our big grape growing region, and we're trying our hardest to find spotted lanternfly as soon as it would ever appear in Erie. So every year we do a survey that I call the Erie Intensive Survey. It's a week-long survey effort towards the end of the season, whenever the adults are active, that we spend up in Erie County. We grid out the county into about 6 different blocks, and we have 2 team members per block, and then sometimes more if we can get additional help. But we do a full-scale survey effort of the entire county to try to find if there are any spotted lanternfly there. And these are our results from this previous year's survey in October. So we went from September to October and completed about 7,262 surveys in Erie County. All right, and then so another duty of the survey crews is trapping. So some strategies, these are very similar to the survey strategies, but one of the main things we're trying to focus on is prioritizing the trapping in unquarantined counties, getting those in the more northern counties in the state to try to find spotted lanternfly, when it first shows up in those counties, using them more as a detection method in those areas. But we also still do trap within the quarantine just for population monitoring. So we have 2 different traps we normally use. We have the circle trap on the right and the bug barrier trap on the left. The bug barriers, we focus more on the unquarantined areas because they're better for early detection. It can catch the nymphs easier and all life stages. And then the circle traps are they're better at catching the adults, and that is we reserve those for within the quarantine. So these are our trapping numbers for last year. We had a total of 321 traps statewide and about 17,000 SLF captured throughout the state. And you can see from the map that we focus more on the northern end of the state with the unquarantined areas, but still do population monitoring in the infested areas. We also this year did egg mass traps. So we put these up in areas where we had higher populations of spotted lanternfly to see how many egg masses we could collect. With these, we don't use the tar roofing like USDA does. We use a sandpaper. It's about 120 grit sandpaper that comes in these big rolls. We use that because of the egg masses getting sent. A lot of them get sent to Melody Keena for her testing, and it works better having the sandpaper instead of the asphalt roofing. But we had a total of 77 traps throughout the state in different latitudes there, and we had collected about 465 egg masses total. And surprisingly enough, we caught a lot out here in the West. I was really surprised this year because in previous years, we didn't catch as much, but I think it was a timing issue that we figured out now. All right, so another duty of the survey crew is public report responding. So some strategies for this, we check the reports daily upon starting the workday, and then we highly prioritize reports outside of the quarantine. So every report that comes in outside of the quarantine is responded to. Now, I noted there that response is not always a site visit. Response can just be calling the report back, but we want to respond to everything outside of the quarantine. And then we can respond to ones inside of the quarantine as necessary, usually if they're outside of known infested areas. But every report is fully investigated if there's a reasonable suspicion of spotted lanternfly. So we call to verify, ask the reporter questions to see if they actually saw what they're describing or what they're describing as spotted lanternfly. And then especially it's helpful whenever they submit pictures as well. So these are the public report numbers from the previous years. So we have the whole way back to 2019. And so you can see it was substantially larger amount of public reports in 2019 compared to 2024, but they seem to fluctuate up and down as the years go by, since we had less reports last year than we did this year, and then the least amount of reports in 21. But with our crew, we responded to every single public report that was outside of the quarantine in 2024. All right, so now we're going to talk about the treatment staff. So the treatment staff, they do a lot of things, but this is kind of it boiled down into three distinct duties. So they do site assessments, which is consenting the properties for treatment, and then also inventorying those properties, the tree of heaven that are on site. And then they do the treatment, and then also quality assurance after the treatment is completed to make sure that everything was completed properly. Excuse me. So for our treatment strategies, I probably should have ordered this a little bit differently, but first and foremost is our prioritization score. So I'm not going to go too in depth on what that is, but it's based on the high-risk pathways, storage facilities, shipping facilities, other things like that we prioritize high because of their risk of spread. And so we first give a property a priority score, and then it gets put into a priority area, which I'll talk about on the next slide, which has the areas with our highest priority properties are within those areas. And that's where we focus a lot of our treatments. And then for the treatments themselves, we treat each property 3 times. The ideal goal is to get 3 treatments of bifenthrin on each property each year. That's not always completely attainable, depending on logistics, weather, things like that. But we try to go for 3 treatments of bifenthrin and 1 treatment of dinotefuran on each site. And for those who don't know, bifenthrin is a contact insecticide, so that is spraying the bugs themselves directly, and then dinotefuran is systemic, so it is spraying the tree of heaven. All right, so these are the priority areas that we have for our different parts of the state, and you can see we do things a little bit differently in each one, but we still use the same score and group the properties together in what I call these little blobs that you see on the screen. But these are areas with the highest priority properties in our zone, and these are the properties that we have signed up fall within these blobs. So we focus on completing all the properties that are within these areas every year, and those can change too. A note there. So this is our treatment numbers for 2024. So of total sites we had assessed, we had 1,284 sites assessed. Signed properties, we had 847. And then acres signed, we had about 45,627 acres. And then for the properties treated, out of those signed, we had about 520 treated. And then a total of 28,393 acres treated. Now you'll see some of those dots are still red. The green is treated, the red was not, but a lot of those were properties that were signed up at the end of the treatment season, so after we could do anything at those sites. And so we have updates for 2025. So some kind of new things that are going on, things that are going to change a little bit. We did finally get tree injection equipment that we can use, so we don't have to outsource that to a contractor anymore. And so hopefully we can get to using that this year and be able to treat some of the sites that are too close to water, which we cannot treat with the other two chemicals. We have a new power sprayer for the central zone, so that brings us to 3 hydraulic power sprayers per zone, so we can get a little bit more done there. We are fully staffed at the moment with our year-round staff. And then also we're looking at eventually adding a technician position for Erie County, since that's our highest priority area. And that would mean that we would have 3 year-round staff assigned to Erie County. Right now, we currently have 2 year-round staff there, but I'd like to get 3 up there so we can have eyes on that year-round with multiple individuals. Also of note, in Northeastern Pennsylvania, we're working with DCNR to survey and treat one of their new land acquisitions. They recently acquired some land that we found spotted lanternfly on, so we're sharing information with them, providing where the tree of heaven are on their property, so they can do with that what they will. And then we are they're providing us with any information they have, and we're going to treat the land that they acquired. And then also in the West, particularly, we're using a lot of herbicide recently to downsize the amount of tree of heaven on our larger properties. Some of our properties have, thousands of tree of heaven on site. So we're trying to get that down to a more manageable number. And we're prioritizing getting rid of trees that are in areas that are harder to access for us with our equipment. So we're trying to downsize that to easier access so we can get things done. And then also we are in the process of renewing our agreement with Norfolk Southern to be able to get back on the rail yards for this year's treatment season, since those are very high priority areas for us. And then that is it for my update for our operations. And I can stay on for questions or whatever works for you, Jacob.
Fantastic, Zach. Thank you. Glad to see everything that's going on. There is a question from Huping in the Q&A section if you want to type out a response. But we are going to move on to our last presentation of the day. Running a little behind, but hope everybody is able to hang on a bit more. Our last presentation today will be presented from the Penn State and Cornell team that are looking at the worst case scenario of what will happen if non-sprayed vineyards get harvested with a mechanized harvester. So the team presenting this will be Brian Walsh from the Penn State Extension and Jen Russo and Megan Luke from the Lake Erie Regional Grape team. Everybody's ready? Over to you, Brian.
Cool. Can you hear me?
We can hear you.
All right. So good afternoon, everybody. I got the pleasure of holding you up from getting out of this today. So I'll try and go quickly. Just real quick, this is a collaboration. The Lake Erie Regional Grape Program is a collaboration between Cornell and Penn State. kind of unique in the extension system. And with me are Megan Luke and Jennifer Phillips Russo, who are of that team. What we took a look at was what happens with lanternflies in vineyards when the grapes are mechanically harvested. And we'll show you a little bit of what we saw happen, what we didn't expect to happen. But the background, the current situation of lanternfly in the vineyards, in the Southeast region, we've seen consistent influxes into the vineyards in the fall, during the adult stage, particularly year after year. And in those Southeast vineyards, almost all of those grapes are southeast of Pennsylvania. Almost all of those grapes are grown for wine. And as such, the blocks tend to be smaller. And even where we have seen the peak population collapse, if you've ever heard the term collapse, lanternfly is collapsing where it started. I would not use that word collapse. I would say that the population has decreased greatly from what it once was from the initial build, but they still exist. And what we see year after year is that there are some areas that will surge back up in population numbers, particularly in the fall. They manage to find their way into the vineyards consistently. Now, it's not uniform across all the areas, and after the big surge and the drop, it tends to be a little bit more spotty. But the additional insecticide applications that are required to control those lanternflies in the vineyards doing this for wine, the vineyard growers in the smaller blocks can do it, and it's also done to prevent the continued feeding by the adults on the vines draining sap. As Flor showed us, the nymphs are not as much of a concern as the adults. The adults, when left go, can really damage the vines and inhibit the overwintering capability of the vines. So the Lake Erie region, as Zach just said, is a particular concern for Pennsylvania and New York. A lot of that's grown there is for juice grapes, and they command a much lower dollar value than wine grapes in general, which means that there are tighter costs associated with them for the growers. They don't have the luxury of just simply adding more and more sprays in that some of the wine growers can do. And that may easily put some of the growers in the juice grape industry out of business if they have to match the numbers of sprays that the wine grape growers have been doing in the southeast part of the state. So what we looked at with the harvest methodology is that in the southeast, it's predominantly hand harvest. They harvest into small bins, they collect them and press in small batches. And what we see in the Erie region, there's approximately 32,000 acres of mostly predominantly Concord grapes, some Niagara, but juice grapes also used for wine, but it's predominantly done with mechanical harvesters. And those mechanical harvesters move fast, and there's no human hands touching the grapes from the vine to being processed. And so there's not all those opportunities to pick out the insects that might get into the harvest. And in general, the juice grape growers don't apply insecticides into the fall. The harvest season ends, and they generally stop with all applications. That's not possible with lanternfly if the adults are continuing into the vineyard. You cannot let them have that free month and a half to feed on the vines unhindered. And so this is a video going back to the Lanternfly Summit last year. Jen Russo shared this, and I'm going to speed it up a little bit, but this is what mechanical harvesting looks like in that region. It's over the row. They're big, fast machines, and they're dumping into big hoppers. And this mechanization, we were wondering what would happen if those vines were full of lanternflies. And the question, and this is what it looks like, not sped up, this is real time, what it looks like on the conveyors. Will the lanternflies will the lanternflies get caught up with those grapes, wind up with the grapes in the juice, in the bins, in the totes? And when the harvester is going down the row, it looks like this in real time. slowed down, this is what it looks like. And so we had hypothesized that the shaking of the vines ahead of the harvester was going to scare off a lot of those lanternflies and get them out of the way. At least we had hoped. And as this continues, right down the row at high speed, we just really needed to see what was going to happen. And thank you to Dr. Terry Bates for sharing this video. And moving on. So what we asked was, what happens if a vine looks like this and then the harvester comes through? So what we did was we found a grower with a mechanical harvester in South Central Pennsylvania who agreed to not spray a block for us, and there was lanternflies there. I'm going to say again, they agreed to not spray that block for this study. This was not sprayed for the study, just to make this absolutely clear. This is not what's going on in a normal situation where the grapes are being harvested with spotted lanternflies present. But when we look out at that 32,000 acres of the lower dollar grapes, we have to know, will they be able to not spray and not have the grapes impacted, the harvest impacted? We're going to withhold the grower's name. We greatly appreciate the donation and the time, and they sacrificed the grapes that were harvested in that block and allowed us to go through them and see what happened. The study was to see how many adults would be harvested with the mechanical harvester, and the methodology was such that the team that was out there, I was not able to be out there that day, but they did a fantastic job. They picked we had 5 rows set aside that weren't sprayed in that whole block. The whole block was not sprayed. And what the group did was they went through, they counted what was present in adults on the trunk, the cord, and the shoots, and the ground. And the grower harvested 100 feet in from the end. And then the bin contents were sorted. They were dumped, and everything was sorted through bit by bit to find the bugs, the pieces of bugs. And they were classified as alive and active, alive, inactive, or dead. And then there was post-harvest counts that took place to see what was still left on the vine. And just so you understand that you have the main trunk, you have the cordon going across the sides, and then you have the shoots. And wine grapes are a little bit different than the juice grapes. The Niagaras and the Concords are much hairier, I'll say, in that those shoots grow a lot thicker. The canopy is a lot heavier. I'll show you what it looks like in the wine grapes in a moment. But those 5 test rows were 100 foot in from the edge line, and then the grapes were dumped into 1.5 ton bins, and it was not good. It turned out to be a whole lot worse than we expected. This is the sorting. This is an idea of the bugs that Dr. Shugart and Tess pulled out of the bins. And what we saw was that on these 5 rows, moving in from the forest edge, that in this case, the first row, 59%, second row, 80%, third, 49, fourth, 66. Last one was 29%, not as bad. But that's the percentage of the insects that were counted on the vines that wound up in the bins with the grapes. So really, really not good. To give an idea of that, the numbers that were in that 100-foot test strip, there was 437 lanternflies. 260% of them wound up in the bin from the first row, and on down, you see. So an average of 510 were counted in each test strip, and representing 22 lanternflies per vine. Not outrageous when you've seen how bad it can get in a vineyard, but an average of 289 were harvested into the bins, and that represents about 60%. So what does that mean? I'm not going to show you the close-up gross pictures, but you can clearly see in that bin of squished grapes, there are squished and live and pieces of lanternfly. Again, I want to repeat, this block was not sprayed pre-harvest specifically for this research, but we can clearly see that the grapes are going to wind up in the harvested crop with mechanical harvester if they're present there in the vines. The percentage of insects represents unacceptable foreign matter contamination that's dictated by USDA. And that includes everything, all kinds of bugs, sticks, leaves. There's foreign matter rules that you cannot exceed. Just a several as low as 2 lanternflies in a 1,000-gram sample would exceed that and cause potentially a 22-ton bin of lanternflies to be rejected at the from the processor. The processing goes on. Those 22-ton loads are getting dropped at the processor during harvest about every 13, 14 minutes. So it gives you an idea of the scale and how fast this is going. There's not time to pick out all the bugs that are present in there. This is raw data, and we're going to keep working with it, but we really need to accelerate the research before lanternfly gets to the Erie region. Since that took place, this was in October of 2024 that first initial study took place. Since then, we've worked on the SCRI to address this, an SCRI grant. We've worked with the state and federal agencies with updating them. We've had research planning meetings with mitigation strategies, as well as with the food science researchers, because we are talking about foreign matter contamination. As Flor was talking a little bit about, there are greater concerns beyond what Flor spoke of, because this is not for wine processing. This is juice that is not going to get fermented. So the question is, will there be chemicals that taint the juice? Is there potentially hazardous chemicals in the lanternflies? And the working groups are in motion with more outreach events on this. And we've continued to work with Dr. Jason Bobin from SUNY Binghamton, who's been working on an AI detection model. And Dr. Bobin was present last year at the Lanternfly Summit in Harrisburg, and he asked for images of lanternflies in all stages. And I have to say, he was very pleasantly surprised and received over 10,000 images from the attendees of last year's summit. And they've begun to develop that AI model. And this is camera footage that was taken during the study, GoPro cameras that were stuck on a harvester for this particular study. And this is what it looks like in real time going along that canopy. And you can see, after it was run through the AI model, you can see the adult clearly picked up, and all these little blinks that you see are where the model thinks it's picking up lanternflies. And this is real time. When we slow that down a little bit, we can see that it's picking up a lot of things that aren't lanternflies, at least from this view of the canopy. And this is wine grape vinifera canopy that's very much thinner than you're going to see in the Concords and Niagaras. The Concords and Niagaras, you generally cannot see through it all. It's so hairy, and with on the vine, the vines, I should say, are so hairy with the shoots. But this is an idea of what it looks like. And when that footage was applied to the conveyor inside the harvester, this is the real-time speed. And so trying to see what's going on with just human eye is very difficult. But the algorithm is able to look at that, we're hoping. And we're hoping that at some point we'll be able to develop the capability for growers to know if there's lanternflies being harvested, that they could stop if necessary before potentially harvesting contaminated loads. This is what that conveyor, same exact footage, looks like, slowed down greatly. And you'll see that the AI can start picking up the individual insects, and it doesn't do a great job, but it is picking up a number of them. It's a first initial try. It's not perfect by a long ways, but it's in the development. And so these are the kind of things that we're working on to help the growers mitigate the concerns. We also worked with Dr. Bobin in a vineyard in Berks County. This is not far from where Lanternfly began originally, and he brought in a drone with extremely powerful camera on it that they are using to see if they can detect egg masses, count egg masses. Ultimately, the intention is to have that software available for roadside scanning with small drones to get an idea of populations present. And in this particular vineyard, we did this one small block representing 1.3 acres of the entire just over 18-acre vineyard. We did hand checking. Liz Dieter and I from Dr. Urban's lab did 3 rows crawling through what was remaining of the snow while they flew their drones. And in those 3 rows, we counted a total of 1,594 egg masses present, including the posts and the vines. This is an area where the population overall has greatly declined, where people talk about collapse. Clearly, there's been a resurgence in the 2024 season in this area of Berks County. There were a lot of adults present there, and the grower had backed off spraying from what they had been doing in the heydays there of 2019, 2020. I shouldn't say heyday, but the worst of the population spikes. And so what these numbers indicate, this is a picture of the big drone flying with the camera down these rows. What it represents is from our physical sampling crawling around on the ground, it was 8.7 egg masses per vine, average of 10 egg masses per linear meter. And the end post, and that excludes the end post. The end post, we know that they like to lay their eggs on. There was average of 46 egg masses per post, and that's not including what had been already scraped off by some of the employees of the vineyard. So in that 1,594 that we counted in those 3 rows, just those 3 rows, assuming 30 eggs per egg mass, it works out to about 47,800 eggs. Assuming a 70% hatch rate, which is not out of the range of possibility, 33,474 nymphs can be expected from just those 3 rows. If we extrapolate that out across the vineyard, sampling indicated 16,900 egg masses per acre, or about 508,000 eggs per acre, assuming 70% hatch rate, 356,000 nymphs per acre. If we take all that out to that full 18 acres in that vineyard, it would represent about 6,478,000 nymphs. Now, we're going to go back. We only did a small block of sampling. We're going to do some more randomized sampling through the other blocks and see if those numbers hold up. But what we saw there, just in those 3 rows that we had sampled that day, this is not gone. This is not collapsed. It is a continuing threat for the grape growers. The lanternflies will come back into grapes when the populations are present. And overall, lanternfly doesn't look like it's going to let off grapes. So with that, this is the field study team from particularly the urban lab. We have Dr. Shugart, Pescavini, and Liz Deacher. From Cornell, we have Jen Phillips-Russo and Penn State. We have Megan Luke, Penn State Extension, Tim Abbey, Dr. Urban, and myself. That grower, really all the growers that we work with, we don't want to assign, we don't want to attribute anything that's negative to them. So we keep their names quiet, but we can't do this without their donation of time, land, and sometimes dumping bins of grapes. And this work was made possible with the funding from the Pennsylvania Department of Ag. So with that, that's what I got.